Let's stay on your lab's work for a moment on the next question, and that is that, how do you express toxicity in these animals? I mean, how do we know that something is toxic to a group of animals?
Right. Well, the key thing in science is to have controls. You need both positive and negative controls. And so in our case, the baseline, or the before the spill, are what we're comparing back to. Or if we're bringing in water, say we're doing a bioassay in the lab, we do embryo exposures in fish, and we're taking water in real time from the coast and comparing it. So then we'll have just a laboratory water that we're comparing those controls to. So you're basically saying it's the percent affected compared to controls. And then for a positive control, you can use just pure oil or constituents that are in that oil where you really know what the effect should be, so that you can sort of see where in that continuum your organisms fall.
Mm-hmm. Anything to add to that?
Well, I guess the one thing that I would like to add is that it's not really accurate to say is something toxic or is it not toxic. Toxicity is entirely a relative term. We have a saying that Paracelsus's law, the dose makes the poison. Anything is toxic at a high enough concentration, even water. And at a low enough concentration, there's nothing that can cause toxicity. So the toxicity that we'll see to different organisms will and is depending entirely on how much oil they're exposed to, dispersed oil, and how long they're exposed to it for, as long as, different life stages, things like that. But it's the amount of dose that they get and the time they're exposed to it that's really going to be the prime determinant of the effects that we see.
Well, are you looking at mostly the toxic effects on individuals or on populations or subpopulations?
Well, so, initially we'll start out by looking at individuals. Initially we'll do what we call a kill them and count them test. We'll try to establish what concentration is required to cause overt mortality in our species. And that's not really because we expect that concentration will be reached in the environment, just that it will then guide us in picking other concentrations that might be more relevant, because we don't have very good data as of yet on actual concentrations in the water that the animals are actually living in. So we'll do that first, and then we'll do a series of other assays. We'll look at individual responses, but we always try very hard to tie it into an organismal or ecosystem level response. In other words, we'll look at reproduction effects. Are the animals reproducing? Are the juvenile forms growing? Are they going to grow up to be able to be reproductive? Are the eggs hatching or are they not? And it's those sort of endpoints that we then try to tie all of the individual level responses, use that into the organismal or ecosystem level.
Right. I think that's what a lot of people's questions are, is what's going to happen to next year's productive stock. Are we going to have, because we have crabs today, we have shrimp today, are we going to have them next year? Sure. So people are very concerned about that. Well, what, we're talking about dispersants, but what about the toxic qualities of oil? Mm-hmm. Either one of you?
Sure. Well, that's what we've been studying prior to this, and the important thing to remember is oil in itself is a complex mixture, and the oil that came out on day one very quickly starts changing. There's some very light or small molecules in there that volatilize very quickly. That while they can be acutely toxic, they're not around for very long. And then what happens is the oil becomes what we call weathered, and then the higher molecular weight, more persistent molecules remain. And so when you're studying oil toxicity, it's kind of tricky because you have to really define what the mixture is that you have. And one of the things we do is pull out individual chemicals to really, again, narrow it down so that we can try and figure out what the mechanisms are. So I think one of the things we do know about oil is it is can be acutely toxic, but then we study fish embryos, and the nice thing about fish embryos is you can watch them develop, right? They're not internal, they're not inside the mother, and we can see one of the big things we see from oil or PAH toxicity is cardiac defects, and so that's a heart, so that their hearts aren't forming properly and they're not beating, and that's obviously going to affect whether they can hatch and then survive. Other concerns, like Joe said, is reproduction, and that's a little bit trickier to measure because it's a complicated environment. Even in the laboratory, it's sometimes complicated to measure. But the impact could be on 3 generations out, so it could be your grandkids in relation to the exposure. And so that's again something we're trying to study in the laboratory with very controlled experiments, but you know, it takes multiple generations to see that, so it's going to take some time. But that's one of the things we think that the constituents, at least in oil, could be doing.
Right. Yeah, I think there's a lot of concern from the Valdez spill about the 4-year lag before the herring fishery dropped, and I think that's got people very concerned down here because people understand it takes a while for science to produce solid results that we feel comfortable with. But everybody's wondering down the road what's going to happen to the fisheries out there.
Sure.
Well,
Any
we're
comments?
going to be monitoring it. I mean, we're going to be
Right.
actively looking at it, and given the available resources, we will be monitoring this on a long-term basis. The Gulf Coast Research Lab, you hear from some of our other people later, have a very long-term database of abundance of different species of fish and shrimp in the Mississippi Sound and surrounding waters, and so we'll be able to say with a high degree of accuracy, I think, what the effect is on the organismal or ecological level based on comparison to that. And you know, we'll from my lab, we'll hope to add in trying to understand what changes at the individual level are driving that response.
Exactly. I think those are questions everybody's looking for answers for. So now we've introduced talking a little bit about dispersants. We've talked a little bit about oil. What about the two together? That always has everybody concerned. Where do we go from there? Is anybody working on that? And if not, when do we get there?
Well, that's what we're actually. I actually brought Christy a sample of the dispersed oil that we made in my laboratory. We got we mix the two under precise conditions, and we make what we call dispersed oil, or the water accommodated fraction. And it's that we're actually using in our exposures in the laboratory because it's unlikely, I think, that anything inshore is going to be exposed to either pure oil or pure dispersant. It's more likely the mixture of the two. The dispersed oil, that's going to be, like you said, much more widely distributed throughout the water column than just the crude oil on the surface of the water is. So that's what we're using in the exposure, and that's what we'll be exposing dilutions of the WAF to different organisms for different lengths of time to look at very precisely what the effects are going to be.
Right.
Yeah.
Anything to add?
Not necessarily, except for that, again, and I think Joe's going to do this, is we have to look at not just whether it's going to kill them, what are sort of the different sensitivities, the different life stages that are important in different organisms. Everybody has a different sensitivity, or every creature has a different sensitivity to these things. Yeah. And so that's some of the work he's addressing.
Yeah, absolutely. That's what we'll all be doing. And I think one of the things to mention is that, it's very, very difficult to extrapolate across species. Sometimes we have to do it, but not all species, even all fish, respond the same way. There can be very big differences in sensitivity, and so we can't test every species in the water. We don't have the time, we don't have the resources, the manpower to do it. So we'll try to pick some species that are representative and extrapolate out. But we have to admit there is some inherent uncertainty in doing that.
Right. Well, in a 2-minute summary of what's happening in your lab. Go back to the high points.
The high points. Well, I think one of the key issues as we go forward is we need the continued funding to keep looking at this, because it could be a couple generations out. And so I don't want this to be sort of a flash in the pan, and really use this, we're in a university setting, to use this as an educational opportunity to get at, so this doesn't happen again, or if it does happen again, how do we do it better? How do we clean up better? How do we protect the organisms better? And so that's what we need, this sort of recovery, tracking the recovery, but also getting at, how can we use dispersants again? Is there a better way? And so that's what I think all this research is going to help us answer. Excellent.
Dr. Griffiths?
Yeah, I think Christy really hit it. One of the primary concerns is going to be continued funding for research. This stuff is not cheap. And Vitrogen doesn't charge me less when I go and ask them, buy a reagent because there's an oil spill out there, and we have to have certain expensive reagents to do the research and to do it right. And so continued funding that allows us to look at multi-generational effects, at ecosystem level effects, at multiple species is going to be absolutely crucial, and we're hopeful that materializes, but we'll have to wait and see.
Well, excellent. Thank you for being with us.
Thank you.
Erin?
Thank you.
All right. Thank you so much, Dr. Thomas. Now, one thing we all think of when we hear about the Deepwater Horizon oil spill is the impact on marine life in the Gulf. Scientists from the University of Southern Mississippi working at the Gulf Coast Research Laboratory in Ocean Springs and the Stennis Space Center in Hancock County have been studying the effects of the oil spill in life in and near the Gulf of Mexico. We talk with some of these scientists and researchers on the coast.
Oh, I think there are a number of issues, including the seafood safety issue, which is being addressed by FDA, by DEQ, state DEQ, those people are conducting a series of rather extensive tests to determine the safety of the seafood here along the Mississippi coast.
I think it's critical that our agencies who are charged with those functions continue to do that, so that they give people that reassurance, and I hope that monitoring will continue. The University of Georgia just came out with a study which is almost the opposite of the previous findings, in that they feel that most of the oil is still in the Gulf, and so that is a question. And I think it's important to continue to look. We need to look for answers. We need to understand what's happening to the Gulf, even above the seafood safety issue.
Other sorts of scientific questions are more of the long-term questions. What's going to happen to the fisheries down the road? Certainly you can see in the news what happens when, say, a bird or a turtle gets oiled, but possibly the big damage is on the larvae of the fish and other organisms, and it's very difficult right now to see what has gone on, how much has gone on, and we're really only going to learn the results of that exposure 1, 2, 3, 4 years out, when we see, do the fish come back?
We've reached a point now where 4 months out, from the original spill, they've gotten the well capped. We're starting to look at longer term, larger scale sorts of impacts. And that has to do with, are we going to see an impact over the next few years in the incidence of disease in fish? Are we going to see impacts over the next few years in reductions in growth or reductions in population size that continue? When you start talking about longer-lived species, like many of the fishes that live along the coast, then we may not see the results of the impact for several years.
Actually, the state of Mississippi is very fortunate in that the laboratory has maintained a 37-year database on abundance of juveniles of our estuarine-dependent species. So we have a lot of information that we can use to assess change.
I would like to know, what is the oil doing in the ecosystem? What's it doing in the salt marsh? What's it doing to juvenile fishes? What's it doing to different kinds of larvae, crab larvae, oyster spat, all the various things that grow in salt marshes and in shallow waters.
Biologically, it's imperative that we understand just what happened. And again, that may take time. But the scientific process ultimately, I think, will provide the answers to many of those critical questions that both scientists and the public, our residents, our people, all of us would like to know.